Good morning, everyone, and welcome again to another CBS webinar. And today we are going to give you an update on H5N1. Where are we now? We're going to have a chat with some persons, very important persons about this. Today we have with us Dr. David Swain, Dr. Elisha Fry, Dr. Richard Webby, Professor Michael Osterholm, and Dr. Trump with us today. So whether you're from here in the Caribbean, the region, internationally, I take this opportunity to welcome you to our webinar today. First, let me introduce our first speaker, Dr. Mark Trotman. Dr. Mark Trotman is the Chief Veterinary Officer of the Veterinary Services Department of the Ministry of Agriculture, Food, and Nutritional Security in Barbados, and has worked for the past 5 years in the Public Services of Barbados and the Cayman Islands, developing national animal health and veterinary public health surveillance policies and disease prevention and control strategies. Dr. Trotman received his veterinary degree from Tuskegee University College of Veterinary Medicine in the USA. He established and operated a private mixed animal practice for 10 years in Barbados before entering the public service, first in the Cayman Islands and then back here in Barbados. He holds a master's degree in livestock health and production from the University of London and a postgraduate diploma in public sector management from the University of the West Indies. He is a former president of the Barbados Veterinary Association and the Caribbean Veterinary Medical Association. He was the first chair of the Caribbean Animal Health Network and president of the World Organization for Animal Health's Regional Commission for the Americas. Dr. Trotman, we welcome you.
Good morning.
Good morning, and welcome once more.
Thank you very much. So since the start of the current outbreak of H5N1 highly pathogenic avian influenza in 2021, we've seen an unprecedented spread of the disease in several countries across the globe. This outbreak is unique in that although wild birds have long been implicated in introducing the virus into Barbados, traditionally, once introduced, the method of spread within the country has been laterally through the movement of birds, products, and fomites. In these current outbreaks, there seem to be multiple points of entry from wild birds with less focus on widespread lateral movement, which is making control and eradication especially difficult. Another unique feature is that the virus is being detected in several wild mammals, and now completely unexpectedly and alarmingly in dairy cattle, hence the title of this webinar. Barbados sits directly in the eastern flyway for migratory birds, including waterfowl, and the several swamps and wetlands dotted across the island serve as landing sites for these birds. Through the excellent work of our host, Dr. Kirk Douglas, we know that non-pathogenic avian influenza viruses have been isolated in these birds. We have not identified H5N1 or any other potentially pathogenic strain, nor has there been an outbreak of high or low pathogenicity avian influenza in domestic poultry on record, but we are well aware of the risk. The Barbados Veterinary Services is partnering with the UWI Center for Biosecurity Studies and a veterinary wildlife expert to beef up our wild bird surveillance for early detection of H5N1 viruses in migratory and resident wild birds. And we have updated our National Emergency Animal Disease Preparedness and Contingency Plans to best prepare for this disease. We have also developed GIS maps of poultry establishments and risk sites and are strengthening our surveillance programs. Today we have 4 distinguished speakers: Dr. David Swain, Dr. Elisha Fry, Dr. David Webby and Professor Michael Osterholm, who will provide us with an update on the current understanding of what we now colloquially refer to as cow flu. I look forward to hearing from them and to the ensuing discussions. Thank you very much.
Thanks very much. I just want to remind the presenters to turn on your cameras before you start. Thanks very much.
Thank you very much, Dr. Troutman. As we go on to our first presenter, we'll hear from Dr. David Swain first. Now, Dr. David Swain is the former laboratory director of the Southeast Poultry Research Laboratory in Athens, Georgia, serving from 1994 to 2003. He is currently a veterinary consultant with bird flu, specializing in poultry, exotic and emerging avian viral diseases, influenza, highly pathogenic avian influenza, and avian pathology. He obtained a Doctor of Veterinary Medicine and Masters of Science degrees from the University of Missouri, and later obtained a PhD from the University of Georgia. He has also received key board certifications, including Diplomat of the American College of Poultry Veterinarians and Diplomat, the American College of Veterinary Pathologists. Dr. Swain's personal research has focused on the pathobiology and control of avian influenza in poultry, especially vaccines and vaccinations. Dr. Swain has served the World Organization for Animal Health international committees to update the avian influenza chapters across in terrestrial animal health code and manual. He currently serves as chair of the executive committee for and the Food and Agriculture Organization of the United Nations Animal Influenza Network. He has been awarded several honors and awards in his length career, including induction to Hall of Honor by the World Veterinary Poultry Association for an outstanding contribution in poultry veterinary science. In 2017, the Science Hall of Fame for Agriculture Research Service and the U.S. Department of Agriculture in 2020. Dr. Swain, we welcome you.
Thank you for the invitation to speak today. And I'm going to concentrate my presentation on the bird aspects, principally concentrating on poultry, but I will touch base on several other topics. So first off, I want to just show you a chart and just emphasize that low pathogenic H5 and H7s are the source of highly pathogenic avian influenza viruses. And since the best records have been kept 1959 till now, 48 high path AI events have occurred, and that's based upon distinct lineages of the virus hemagglutinin. Of these, 45 we would categorize as emergent. That means they emerged from the low path predecessor and were eradicated within a year or two. That's 41 of those have been eliminated. We also have 4 that are currently active in South Africa and Australia. But of course, what we're most concerned about are the 3 viruses that are entrenched in various parts around the world. And for us today, the one of most concern in the discussion is the H5N1 goose Guangdong lineage 2344B clade. And that has produced global outbreaks in a variety of animal species. But I want to emphasize to you that we also have to remember that we can have an emergence of a high path virus from a low path H5 or H7 anywhere in the world. And so that still must be on our mind in our examination, both surveillance-wise and diagnostically. So the current goose guangdong virus, the lineage 2344B, really is the fifth wave of the goose guangdong lineage. And the prototype virus really was identified in Central Asia as an H5N8 in the fall of 2020. And this virus moved from Central Asia into Europe, Eastern Asia, down into the Middle East and into Africa. And then it returned during spring migrations northward in 2021. In the some between the spring and the fall of 2021, the virus moved across the North Atlantic, first identified in Canada. And then in the winter of 2022, it moved down the eastern coast of the United States, and spring went northward back through the US into Canada. And then in the late summer of 2022, it moved southward back into the lower parts of the United States. In the fall of 2022, it moved on down into Central America and at least made one contact and detection in the Caribbean, that was in Cuba, and then into northern parts of South America. In the winter of 2023, that virus then moved on down the Pacific coast. And in the spring of 2023, it moved laterally across into Argentina and Uruguay, and then northward into Brazil. In the fall of 2023, the virus moved into the very southern parts of the southern hemisphere with identification of cases in wild birds in the Falkland Islands, as well as the Antarctic polar front, first in the South Georgia Islands. And then most recently, in February of this year, it was then identified in Antarctica in both mammals and birds. So globally, if we look back from the beginning of 2022 up till now, we've had reported to the World Organization for Animal Health 9,243 cases in poultry, of which this is accounted for 27 million poultry that have died, and 314 million have been culled as part of the outbreak control programs. So with the first virus that was identified in Central Asia was an H5N8, and through that travel into Africa and back out, most likely in Egypt, where a reassortment occurred, the H5N1 emerged and has become that dominant strain that has spread across multiple continents in the world. It is through this reassortment, it acquired additional biological features, including the ability to infect a wide variety of bird species, and to date, over 350 species representing 51 families and 25 orders have been reported as far as infections. And in addition, this virus has been spread by the movement of wild birds, both short distance and at least long distance, which is probably a process of multiple shorter movements across large geographic areas, with many asymptomatic infections in our traditional dabbling ducks, but also there have been individual bird deaths reported, and even up to massive die-offs in seabird colonies. In the local areas, many of these birds have been preyed upon, the waterfowl, by a variety of predatory and scavenger species of birds, and they've become infected, as well as spill over into many of our pet animals, primarily dogs and cats, from a direct contact with those infected wild birds and consumption of carcasses. This has spilled over into our poultry from either direct contact of wild aquatic birds to outdoor reared poultry, especially in backyard or hobby flocks, but most importantly through indirect spread by the wild aquatic birds that are infected, depositing feces in the environment around the barns, and that being then moved into the barns by human activity through contaminated fomites on clothing and shoes, equipment, and in some cases even feed and water. But there's also still an acknowledgement that there is some spread farm to farm, and it depends upon the location and the types of biosecurity used in those organizations. And of course, that's through the human activity of production of agricultural products. This virus has then spilled over into more than 40 species of terrestrial mammals and at least 13 species of sea mammals, with large die-offs, first of harbor seals in the US in 2022, and more recently in South America, sea lions and elephant seals. And also reports even in farmed mammals and mink and a variety of furred animals in Europe, but also most recently, which is the focus of this seminar series, will be in dairy cattle in the US. And our next speaker will speak to that in depth. And there's also been sporadic cases, mostly from direct contact with infected poultry, or most recently with infected dairy cattle. If we look at the poultry cases, we can see that there is a seasonal reporting of the cases. For example, in the Northern Hemisphere, when collected data analyzed by the World Organization for Animal Health from 2005 to 2019 shows that the increase in cases usually begin from the baseline around October and usually peak in February. But with the most recent year's data, which is 2023, this has shifted such that that increase has begun in the early parts of September with an earlier peak in November with the current virus. When we look at the Southern Hemisphere, where Hypath-AI is a more recent introduction, we can see that the beginning of cases increasing usually in early June with a small increase and a peak somewhere around September in poultry. So if we look at just the Americas and kind of a summary of what has transpired, what we'll see is if we look at the last semester of last year, July to December, we'll see that there has been a decline in total bird outbreaks. And that's mainly from a decline in the wild domestic bird reporting, that's wild birds and backyard poultry, as well as a decrease in the number of actual commercial poultry cases. If we look at poultry specifically, and we'll see that there has been a decline in outbreaks last semester of last year, but there has been an increased number of culling of birds because of large farms that have been infected. But generally, there are fewer outbreaks than were reported in the previous year for the same timeline. We can say that with 2024, there has been a decline in the number of reports of infections in wild birds as well as backyard poultry and in commercial poultry cases. One of the new introductions and the new issues has been the epidemiology change of the virus, in this case, where poultry outbreak cases in the United States now are becoming linked to the viruses that are part of the dairy cow herd infection process and that movement of those infected dairy cattle. And so many of the cases in the U.S. that have been in poultry beginning in April have been through this contact, through dairy cattle systems. We can say that globally, there has been generally a decline in both poultry as well as the wild bird backyard flock cases around the globe. So as far as control and guidelines, the World Organization for Animal Health do publish two different publications that help in developing programs of control for Hypath-AI. The first is the Terrestrial Animal Health Code, and the second one is the Manual of Diagnostic Tests and Vaccines. Both of these are outcome and risk-based provisions that help in prevention and control the spread of Hypath-AI, including the prevention of spread through international trade, but at the same time limiting and avoiding unjustified trade restrictions. The code specifically contains very specific considerations, and these are part of the control programs for defining a country or a zone, which is a geographic area smaller than a country level that can be free from Hypath-AI based on surveillance provided by that country. But also it has considerations and provisions for free compartments that are those under a management-based system. For example, genetic companies who produce genetic stocks, either as fertile eggs or day-old birds, may have a compartment system recognized by other governments. There's also provisions for the localization of any Hypath-AI outbreaks to a smaller zone within a free country or a free zone. And it provides specific recommendations for the recovery of that free Hypath-AI free status for that country or zone or compartment. The code and the manual both support vaccination as a control tool, and it supports trade in vaccinated poultry in the presence of appropriate surveillance to demonstrate that freedom of Hypath-AI infection. You may ask why and how. Well, essentially, the requirement for surveillance to demonstrate freedom is very similar to that for non-vaccinated poultry to be able to declare a free country, zone, or compartment. But most recently, and that was last December, the World Organization for Animal Health had a policy briefing where it talks about stricter biosecurity measures and mass culling of poultry may no longer be sufficient to control the disease. And poultry vaccination can no longer be excluded from the available alternatives and should be considered a complementary tool in Hypath-AI control. So what are the principal barriers? Well, the trade barriers by importing countries, principally on meat and exports and imports into their countries. And of course, the other issue is defining what is appropriate surveillance needed to demonstrate that freedom from infection in those poultry. So our primary preventative programs involve movement controls from infected premises and stamping out to prevent that lateral spread. We also have emphasis on biosecurity and keeping the pathogen out of those naive flocks and also keeping the pathogen in infected flocks. And then, of course, vaccination to increase resistance of poultry to Hypath-AI infection. So if we look at just the Americas today, there is vaccination programs that are going on in the Americas. It's been an emergency vaccination program really against the H5N1 virus, the Eurasian goose guangdong lineage. And for 2023 and continuing in 2024, there have been 7 countries that have been conducting this vaccination. Mexico, Guatemala, Ecuador, Bolivia, Peru, and Uruguay against the high path AI, very specifically in their vaccination program, but also Dominican Republic does do some H5N2 low path vaccination. These individual countries are undergoing currently assessments to determine if the vaccination will continue or if they'll stop the emergency vaccination program based upon a risk analysis of infections or lack thereof in migratory birds as well as in domesticated poultry. And thank you for your attention.
Thank you very much for your presentation, Dr. Swain. Up next, we have to present to us Dr. Elisha Frey. Dr. Elisha Frey is an assistant professor of practice in the Department of Population Medicine and Diagnostic Sciences at the Cornell University College of Veterinary Medicine. She graduated from the Cornell University College of Veterinary Medicine in 2010 and then practiced mixed animal medicine and surgery in New York for 8 years. She joined Veterinary Support Services at the Animal Health Diagnostic Center in 2018 and successfully completed her American College of Veterinary Preventive Medicine board exam in 2021. Her main area of interest include infectious diseases, public health, and animal agriculture, and the intersection of all three. Today, Dr. Frey will give us an update on the current situation on H5N1 transmission in cows, the impact on beef and the milk industry, and measures to stop transmission in herds and to animals as well. Welcome, Dr. Frey.
Thank you very much. I'm going to share my
Welcome.
screen here. Okay. Okay. Are you seeing my appropriate slide? Yeah, okay. Thanks for the introduction. My name is Elisha Frey. I work at the Cornell Animal Health Diagnostic Center, which is the New York State Veterinary Diagnostic Lab, and I'm going to be discussing H5N1 in the United States livestock from March 2024 to the present. I was going to start with a bit of geography first. This is a map of the United States, and the Texas Panhandle is this part of Texas that sticks northward, and that's where it all began. Texas borders Mexico, and then New York is way over here. Sorry, my arrows are not pointing where they're supposed to be, but so New York is a far cry from Texas, as you can see, and I was working with veterinarians in Texas to get samples sent to our diagnostic lab. This is a map of just New York State, and to visualize it better, New York City is south, and then Ithaca is where the university is located, and we're kind of in the middle of the state. There are a lot of dairy cattle in northern New York and then also in western New York as well. Dr. Swain kind of covered this a little bit broadly, but I was just going to mention specifically for the US, we've had this sustained H5N1 outbreak in poultry from February 2022. That was the first detection in poultry in the US. And as of today, I just checked on the USDA website before this webinar, we've had 1,155 flocks affected, 500 commercial flocks, 655 backyard flocks. 48 of the 50 states have been affected, and we have depopulated 97.17 million birds, which is a huge number, much more than the 2014-2015 outbreak. But to put it in perspective, the US population for domestic poultry is 378.5 million egg-laying chickens, 9.4 billion broilers, and 218 million turkeys. And then, as Dr. Swain also referenced, we've had outbreaks in wild mammals. So this is a map on the right of detections in wild mammals in the United States, and there's a legend on the far right of the different species, and then you can see where they have been found in the country. So we've had 21 species test positive. They are neurologic due to encephalitis or brain swelling, and this is fatal. Pictured here are some fox kits that were hospitalized at the wildlife rehabilitation facility at Cornell and then died and were H5N1 positive by PCR testing. So quite a range of different mammals, most of them carnivores, You can see mountain lion, black bear, brown bear, polar bear, sadly, dolphins, different marine mammals, seals, coyotes, red foxes, fishers, otters, squirrels, raccoons, skunks, and possums. In February of 2024, there were many anecdotal reports from veterinarians and farmers about huge numbers of sick cattle in the Texas Panhandle, with up to 20% of the herd affected at one time. The main clinical finding was a massive drop in milk production, and you can appreciate this cow's udder that is very devoid of milk and kind of flaccid, and this right rear quarter is really deflated. They also had thick colostrum-like milk. So here is a picture of some milk on a California mastitis test paddle. You can see how abnormal it looks and colostrum-like. There was also a decrease in feed intake, so cattle were anorexic. They had clear nasal discharge, pictured here on these cow noses, low-grade fevers, dry and tacky feces, which some folks were debating whether that was just a sequelae of being dehydrated or actually part of the disease process. And then some farms did have fairly low mortality rates, but not zero. Our diagnostic lab received samples from 2 farms in Texas. The first farm to submit samples was milking 35,000 cows, so a very, very large dairy by US standards. They had over 1,200 affected cattle in late February. Initially, they sent us milk, serum, and feces from the cattle. And our lab, like other diagnostic labs, we're looking for the common infectious agents in cattle. And so we had a bit of a limited sample set, and we're not finding anything significant or consistent in the samples. Later, a veterinarian contacted me that was in Texas on a farm milking 4,000 cattle. The outbreak began on that farm on March 9th, and on March 13th, there were 100 sick cattle that day. So a huge number of sick animals to try to manage while also having a functioning farm. I encouraged him to send our laboratory EDTA whole blood, serum, milk, nasal swabs, and feces. And we decided to just jump to whole genome sequencing based on the previous lack of results from some of the other diagnostic labs in our lab on the previous samples. These are photos of the milk we received from the second farm, and you can appreciate how abnormal the milk is. These photos were actually kind of taken in jest because I sent them to the virology laboratory, and they were not happy with me because they were like, why are you sending us this very abnormal milk? But actually the photos have ended up being really useful for kind of expressing to folks how abnormal this milk actually is. The farmers and veterinarians were also reporting other dead animals on the dairies, including birds and farm cats, which we found out later were being fed raw milk, which is a common practice. Cats live on dairy farms and hang out and drink raw milk. And some farms were reporting a complete disappearance of their farm cats. There was also a dead raccoon. This is a text message that I saved between myself and the veterinarian in Texas about the mortality in the cats and the birds, which was starting to kind of raise the hairs on the back of my neck that maybe this could be HPAI. On March 20th, we did H5N1 PCR testing on the grackles and pigeon we received at the lab, and they were positive. 2 days later, we did H5N1 PCR testing on the cat, and that was very high positive in the lung and the brain tissue. And we simultaneously had the results from the nasal swabs that we sequenced, and 2 of the nasal swabs from the cows were H5N1 positive. So we sent those samples to the National Veterinary Services Laboratory, which is the USDA laboratory. That is the protocol when a transboundary disease is found. We have to confirm it through the USDA. The Iowa State Veterinary Diagnostic Lab and the Texas Veterinary Diagnostic Lab also detected H5N1 within 24 to 48 hours of our lab. So all 3 of the labs found the virus. And then on Monday, the 25th of March, the USDA made the announcement that HPAI had been detected in livestock. We then turned to the milk and did PCR and virus isolation on the milk and found tons of virus in the milk, much more than in the nasal swab. Non-lactating animals and young stock did not appear to be sick from the virus. And then there was interstate movement of cattle that were asymptomatic at the time that spread the virus from Texas to Ohio on the 7th of March, and that concluded cow-to-cow transmission was occurring. Here's some just snapshots of the media after the USDA made the announcement. There was a lot of coverage of this in the press. New York Times and PBS and things were reporting on it quite frequently. The USDA put out a publication titled Emergence of Highly Pathogenic Avian Influenza A in Cattle. And I was included in this paper at our lab for the initial detections along with other folks. And then here's a phylogenetic tree of the H5N1 virus in cattle. And then this paper was published at the time when there was just one human case from dairy cows to people. So that's this person here. And then some maps showing the chronology of the spread of the virus out of the Texas Panhandle to the Midwest and then to other parts of the country by mid-April. Recently, our group collaborated with some other diagnostic labs to publish a paper about 9 dairy farms in Texas, New Mexico, Kansas, and Ohio, cumulatively 78,000 cows. Between the different laboratories, we received 323 bovine samples. This is in preprint, but should be fully published soon, within a couple weeks. We summarized the findings and the timing on the 9 different farms. And so you can see here that we have them just numbered from 1 to 9, and then we have the state where they were located, and then the total of number of animals per herd. So these are quite large dairies. And then the number of animals affected by HPAI with a percentage. So you can see that somewhere between 3% and 20% of the herd was affected. And then we have the date of the clinical outbreak. So this was the Texas herd milking 4,000 cattle that sent the abnormal milk in the vials. This is the herd that was affected earlier, but we didn't make the detection until a month later almost. And then the Ohio farm that received 42 lactating cattle via transport and then broke with the virus a couple weeks later. Here, I just want to draw your eyes to, the timing of the outbreak in these New Mexico, Texas, and Kansas herds. So you can see that they are all clustering around the middle of March. So it's quite interesting that this outbreak was really close in timing between different locations of farms. And also, again, very large farms, 4,200 cows, 7,000, 4,000, up to 10,000 cows, with a range in the percent affected, which, you know, this is anecdotal, right? This is if folks are writing it down or recording it or putting it in their dairy comp systems, and kind of what the individual herd criteria is. So I think that this is, you have to take that with a grain of salt, the percentages of affected animals. We looked at shedding patterns in clinical versus non-clinical animals. Initially, the USDA was encouraging veterinarians to test 10 symptomatic and 10 asymptomatic animals when this was still a mystery disease. So the purple dots are the clinical cows or the symptomatic cows, and then the green dots are the non-clinical animals. And then we have sample type here. So milk, nasal swabs, urine, and feces. And you can see that the viral load is highest in the milk from clinical cows, but both clinical and non-clinical cows have virus in less amounts in the nasal swabs and the urine. It appears that feces is not a source for virus, and this has been consistent with sampling at other labs as well. We had a subset of animals that we serially sampled their milk. So on day 3, day 16, and day 31 post-clinical diagnosis, and we looked at the viral load in the milk at those time points. And you can see that there is a fair bit of virus initially, less virus as time progresses, but not no virus. And then when we look specifically at the infectious virus load, we see that at day 3, there's infectious virus, and then by day 16 and 31, there's no infectious virus in the milk. Here we have a photomicrograph of H5N1 tropism for the bovine mammary gland. So at the top, we have a zoomed out view of the histology of a cross-section of mammary gland from a deceased cow that had H5N1. Zone 3, where this blue arrow is pointing, is the normal mammary gland, and that correlates to the bottom row of images. Zone 1 and Zone 2 are abnormal mammary gland with H5N1 virus. And so you can see here, these round circles are fat globules, which are normal parts of milk. And then we have a loss of the architecture in Zone 1 and Zone 2 with epithelial cells sloughing off and cellular debris. This slide panel in the middle with the ISH at the top, that stands for in situ hybridization. And so that is a staining method for the influenza A matrix. And so where we see this dark red in zone 1 and zone 2, that's all virus being detected, influenza A virus being detected in the mammary gland. Similarly, for this final panel, we have immunohistochemistry. So highlighting dark brown to gold is where the virus is in the mammary gland. And then if you look at the normal tissue, you see a lack of staining for ISH or IHC. This diagram is a model of the spillover and spread of HPAI H5N1 genotype B3.13 in dairy cattle. As Dr. Swain kind of described, we had clade 2.3.4.4b in circulation, and then a reassortment event occurred, resulting in genotype B3.13, and then some detections in wildlife in early 2024, and then an unknown event, and then a spillover event into dairy cattle of B3.13, and then a spread from the dairy cattle to other species, domestic cats, raccoons, birds, and then intraspecies transmission to other cattle, which then spread it to other facilities, including poultry facilities, other dairy farms, and 3 individual cases of human disease. In response to H5N1 in cattle, the USDA mandated a federal order effective April 24th of 2024, which required interstate movement of lactating dairy cattle to have a H5N1 PCR performed on milk samples. This federal order did not include non-lactating or dry cows going directly to slaughter. So cows that are crossing state lines but going direct to slaughter do not need to be tested. And young stock are also exempt from this federal order. This is an image on the right of milk samples coming to our laboratory for the H5N1 PCR per the federal order. Official testing of H5N1 can be performed at a NALN laboratory. NALN stands for National Animal Health Laboratory Network. And there are 42 NALN laboratories in the US, and these are laboratories that the USDA has approved to do NALN testing. So here's us, we're this green triangle in New York, and all of this testing is paid for by the USDA, so it is free to the farmer. All NALN laboratories and private laboratories that are doing this testing are required to report to the USDA if there is a non-negative result, and then the USDA will rerun the H5N1 PCR and confirm it, and then it is called a positive. So that's the protocol for transboundary diseases in the US. This is a current map of the US and the status of H5N1 in livestock. So we have 129 total cases, which would be the same as 129 farms. So these are not individual cows, these are farm numbers. We have 12 states affected, and then the shades of blue correlate to the number of affected cases or farms. And so you can see that it is kind of centered in the middle of the country, but it is slowly starting to spread. Per the USDA, they have tested over 17,500 bovine milk samples since the federal order in April of 2024. And just for a frame of reference, there are 9.36 million lactating cattle in the United States. This is a picture of a typical dairy parlor on a large farm. You can see here there's employees in the parlor attaching the milking units to the cow's udders. And so you could see that this would be a place where aerosolization of milk would be occurring, and also a lot of animals in a tight space with nose-to-nose contact with each other, and also, getting contact with milk as well. So cows and humans in contact with milk, which we know has virus in it. If we look at just the testing done at our diagnostic lab, which is one of many NALN laboratories, we have tested 2,421 milk samples per the federal order. 2,376 of those samples were just for movement or screening purposes, and then we have tested 45 samples that were sick or H5N1 suspects. On the right, I have a graph of the number of samples tested per day, and you can see this big spike here on the 31st of May, and that was because one farm in New York State tested all of their cows. They were selling them all. They were having a dispersal sale, and they wanted to test all of them before they sold them. So that was over 1,000 samples that day, which is actually a good proportion of the total number of samples we have tested. On the bottom, I have the states listed where we have received samples for testing. This is mostly the Northeast where we are located. I wanted to show some images of lab samples moving through our laboratory. I thought that might be kind of fun for folks to see how we do it. So first, we have our veterinarians submit the samples in boxes with Fs on the outside. They also have to email us so we know that they're coming. This pathogen has just been removed from the select agent list temporarily for 3 years, but we are still using PPE or personal protective equipment when handling these specimens. So they have to get racked and labeled and put into our computer system. And then they go up to our molecular diagnostic laboratory where sample extraction is performed. And a master mix preparation is added, and then they are loaded into the plate. And then we have a 96-well plate that is put into our PCR machine. And then the PCR results are put out for the client. And you can see here, not detected means that we did not detect any of our target or any of our influenza A H5N1 virus. And the CT value or the copy threshold is undetermined. So the threshold for PCRs is set, and then we start cycling, looking for our target. And once the target is reached, that correlates to a cycle threshold, which is the number of cycles. So the lower the cycle, the more target. If the threshold is never crossed and the line stays below the threshold, that is read out as not detected, and the CT value is undetermined. So what you want to see is not detected or undetermined. But if there's a positive result, it will cross the threshold at some point. Briefly, other H5N1 testing and information, the USDA will also pay for other testing beside the milk in the federal order. So you can submit nasal swabs on non-lactating animals. We know that they don't shed as much virus in the nasal swab, but it's the other sample that we can test in non-lactating cow. Dead cats and birds on farms can also be tested at no charge. And recently, the USDA rolled out an H5N1 dairy herd status pilot program, encouraging states to enroll farms. If they test their bulk tank weekly for 3 weeks and they are negative for H5N1 each time, they are considered a monitored unaffected herd. And then they can ship animals interstate without individual cow tests, but they have to continue to test the bulk tank on a weekly basis. A very small number of states and herds have enrolled in this program. It has not been super widely accepted, but it's still in the early stages. The USDA is reimbursing for lost revenue for farmers. I am not directly involved in that reimbursement, but it is stated on their website, and I know that some veterinarians are pursuing it. It is even retroactive back to the first cases. There have been 3 reported human cases of HPAI from contact with dairy cattle in the US since April of 2024. 2 dairy farmers just developed conjunctivitis or pink eye. This is a picture of the first person who developed pink eye from H5N1. He was in Texas, or she, sorry, actually, I'm not sure. And then the other 2 employees have both been in Michigan. The most recent human case developed cough and fever, some more of the typical influenza-like symptoms. There has been no human-to-human transmission to date, and all 3 people received an antiviral commonly referred to as Tamiflu. Thank you for your attention. That is all I have.
Thanks very much, Dr. Frey. I want to, before we start with Dr. Webby, to just remind the audience to complete the first poll that was launched so that we can get some really important data for ourselves. So over to you, Asanchi.
Thank you. Okay, I want to say a special thank you to Dr. Frey for her presentation. Our next speaker will be Dr. Richard Webby. Dr. Webby is a member of the Department of Infectious Diseases at St. Jude Children's Research Hospital in the USA and the current director of the World Health Organization Collaborating Center for Studies on the Ecology of Influenza in Animals. He received his PhD from the University of Otago, New Zealand. Dr. Webby has a basic research program funded by ALSA, the fundraising arm of St. Jude and the U.S. National Institutes of Health that focuses on influenza viruses at the human and animal interface. His work involves virologic and serologic surveillance activities in animal and human populations to determine the prevalence of influenza viruses present. Dr. Webby has several distinctions and honors, including the Special Recognition Award in 2010 in honor of extraordinary work in H5N1 influenza research with the National Institute of Allergy and Infectious Diseases, Division of Microbiology and Infectious Diseases. He is an expert group member with WOAH and FAO swine influenza, and a member of the Influenza Advisory Committee with the U.S. National Court Board. Today
Excuse
he
me one
was,
sec,
sorry?
sorry. I'm sorry to interrupt here, but I just remembered, maybe we should switch the order for Dr. Webby and Professor Osterholm, because remember that you guys are hard stop at 12.
Could Dr. Douglas, could I suggest that you keep the order, because I'm commenting on some of the comments that Richard's making.
Oh, okay. No
So
problem.
I'd be happy to follow him, okay?
Okay, I just wanted to
Thank
make
you.
sure.
Thank you.
Okay, no problem.
Okay.
Thanks.
So today Dr. Webby will speak to us. He will speak to us on H5N1 and bird flu. What did the gene say? Welcome, Dr. Webby.
Thank you very much. And Mike,
You're welcome.
I'll try to get through this so you don't miss your next appointment. So what I'm going to do is basically work through a little bit more and put a bit more meat on the bones about what we know about the specifics of these viruses, particularly the 2344B, which we'll explain a little bit more about what that means, and then sort of home in on a little bit more about these viruses that are now infecting the cows, what we know about them, their makeups, are they changing, is there anything specific about those viruses? So we've heard basically David's presented a much more in-depth overview of this, but thinking back to when we first saw these H5 viruses, the so-called goose guangdong lineage of H5, the so-called name because the very first isolation we have was from a goose in Guangdong province way back in 1996. And we've seen all these waves that David talked about as these viruses started their life off in Southeast Asia and then started to explore different geographic regions, different hosts, of course, to where we are today, where most regions of the world are impacted by descendants of that original virus detected in southern China back in 96. And again, just to reiterate, I know this is probably old information from most, but flu viruses evolve in a couple of major ways. One, like all organisms, through mutation, so just point mutations in their genome. Flu is a negative sense RNA virus, so of all sort of the various shapes and shapes of viruses out there, it's these negative sense viruses which have the highest ability to evolve through mutation. So they make a lot of mutations every time they replicate. Some of those may have phenotypic impact on the virus. And of course, the other one, which we've heard referred to a number of times already, is reassortment. This is a property of the fact that the flu has its individual genes on individual pieces of RNA. So two different viruses get into the same cell. And what comes out is sort of a whole soup of viruses, the parental viruses or viruses with a mix of gene segments from either of the two parental viruses. And we're at a point where we can't predict reassortment, and I think even more worryingly, we can't actually even predict the phenotype of viruses that come out of this activity. Sometimes these viruses that come out with a unique combination of genes actually have biologic properties that neither of the two parental viruses had themselves. So it's a way that this virus can really evolve in leaps and bounds. And it has been a hallmark feature of the evolution of the goose guangdong lineage of viruses, particularly the 2344Bs. So what do we mean by the 2344Bs? Again, just to remind people of what that is. As I said, the flu virus evolves through these point mutations. A lot of those occur within the hemagglutinin, so this H5 part of the virus. Over the course since 1996, a number of mutations have occurred, and this is just a phylogenetic tree of the HA. This is just a snapshot since about the early 2000s on. Actually, these viruses continue to evolve over time. Some of these lineages have been replaced. So if you even come to the current day, well, current meaning, I guess 2021, 2022, and 2023 as well. A lot of these earlier lineages, so the 2.2, the 2.3.2 ones, at least some of the 2.3.2 ones, and lineages before, a lot of those have actually disappeared. I don't think we have to worry too much about the numbers and letters here, but these are a sort of a phylogenetic tool that we've used to basically communicate this diversity of these viruses. It became apparent early on during this outbreak that there was a need to communicate which group of viruses we were talking about. So sort of a joint effort between, well, FAO and WHO came up with this nomenclature system. Again, it's based on the similarity of viruses within these clades, similarity between clades. And when these
Dr.
viruses...
Webby,
Yes.
we're not seeing your slides.
Oh, that's not ideal. Oh,
Sorry to interrupt you, but I thought you might want to show them because I'm sure you worked on them.
yeah, sorry, first time doing Zoom. No. There we go. Sorry. Now you should be seeing something, correct?
Yes, we are.
Yes, thanks. Sorry about
Yeah.
that.
Oh, that's what I talked about earlier, so we can forget about those. Sorry, this is a slide I was on. Apologies for that. I was talking about here. Again, I won't bore you all with the same details, but this is the sort of phylogenetic tree, the HA I was talking about, looking at some of these earlier lineages have disappeared. So at any given period of time, a certain amount of diversity circulates. New versions come on, some of the old ones disappeared. Right now, if we look at the diversity of viruses circulating now, we have the 2344Bs, of course. And we also have some of the old 2321. So 2321As hanging on in parts of the Indian subcontinent, and the 2321Cs in parts of Southeast Asia. The other part, of course, reassortment. Reassortment has also been a sort of classic part of the evolution of this lineage of virus. And again, this here is just to show a little bit of what happened once these 2344Bs got into the Americas. So to walk you through this a little bit, this is just a percentage of the total number of sequences pulled out of both sequence we've generated and from the public domain over a given period of time. Each color here represents a particular combination of gene segments. Or genotype. And you can see if we, even since the early days, these viruses first came into the Americas as a Eurasian genotype. So at least early on, a single introduction. There have been multiple since. But as soon as these viruses got in the US, they started to interact with North American viruses, mix and match gene segments, and we saw a sort of a soup of different combinations of genes along the way. At some periods of time, there were multiple genotypes circulating. Other times, they were more dominant. And you can see that the pink one here towards the end, or pinkish, represents the B313 lineage, so the specific genotype of virus that jumped into cows in the US. I should note that this particular combination of genes wasn't actually dominant at all, at least in the available sequence information leading up to this event. There wasn't a whole lot of activity going on in birds prior to this event, and certainly in the samples we received from our collaborators in Squidus at UGA from wildlife. Again, this particular combination we didn't see, but it's now making up a large percentage of at least sequenced isolates. That's because, of course, there's a lot of sequencing going on in this area now. If we come back to sort of ask the question, what else are the genes telling us about this introduction? Dr. Fry sort of touched on this a little bit. This is just again from the paper that she mentioned from USDA and a lot of the collaborators as well, looking at the specifics of the virus that made this leap into cows. In the blues and greens here, these were the viruses that were circulating prior to the outbreak in cows. The B36 was a fairly dominant lineage genotype of virus, at least circulating in some of the wild birds. It picked up the PB2 and NP, either directly or through some intermediate. And then that particular virus was the one, that's that B313 genotype I talked about, made it to cows. And looking back, sort of the experts at looking this, and a number of people have done this, you come to the conclusion that this is a single introduction. So all that activity we're seeing in cows now, at least from a virologic perspective, looks to be a single introduction and then spread within cows from that single crossover event. I think the other take home from the studies is the fact that this crossover, although we didn't detect it until March, probably happened sometime late 2023 to early 2024. So this period of time where this virus was presumably circulating in cows but undetected until March. So again, two things: single introduction. So these viruses, I think this is important for other parts of the world as well, and also the US. This is a fairly rare event of these viruses crossing over into cows. So it doesn't seem to be happening. Well, I guess at least we have not detected this bird to cow spillover frequently at all. And this is perhaps a single introduction we know of. So not a common, I guess, host switching of these viruses. But when it has made that leap, of course, it's been very, very successful. From the other point of looking at the genes of the virus and what they can tell us, from a human health perspective, of course, we all want to know what the risk to humans is. As Dr. Fry mentioned, we've seen 3 confirmed cases. I think everyone is aware that the number of real infections is probably substantially higher than that. But from cow to human, 3 confirmed cases, luckily no evidence now of onward human spread. And that's sort of consistent with the goose guangdong lineage of viruses over the past 25 odd years. There's 800-900 human infections with this goose guangdong lineage of viruses. Some suspect of some limited human-to-human transmission, but certainly nothing. Nothing a major, of course, that if something did change, then we are talking about a pandemic, and that's a place we don't want to be with this particular virus. And so what do we know about the genes or the changes? Again, I'm not going to go too much into the specifics here. I like to sort of break down the categories of changes. So what's it going to take for this virus to change from being a bird or a cow virus today to being the sort of fully fledged human pathogen tomorrow? We know there's receptor binding, so all influenza viruses at least bind to sugars on the surface of host cells. The avian forms of this virus likes to bind those sialic acids in a terminal 2,3 conformation. Human viruses like to bind that in a 2,6. And we know the specifics on the hemagglutinin of HA that interact with those sugars. So we know at least some genetic signatures to look for. We also know that for these viruses to switch hosts and become more adapted to human, they've got to, overcome some of the innate responses in the human cells, and when they get into the cell, then replicate within the context of a human cell. So interact with human proteins inside that cell as opposed to avian or bovine or fox, or pick your favorite animal. And the other sort of HA stabilizing mutations, this we know is important, and that transition from being an avian virus to being a human virus does seem to also look for some sort of stabilization of the HA, particularly as related to pH changes. If we look at what we've seen in humans, not humans, if we look at what we've seen in these 2,3,4,4b viruses, sort of irrelevant of which host they've been in, we haven't seen much to suggest changes really occurring on the receptor binding. HA stabilizing, I think we still need a lot of work in that area, but no, at least smoking guns. We have seen some of these replication changes, particularly when we look at the mammalian hosts that have been infected with these viruses. Where we have seen, particularly in the polymerase proteins, some changes that are associated with an increased ability to replicate in mammalian cells. So these are, I like to see, the sort of the low-hanging fruit for these viruses as far as the I guess, flu viruses in general, this is the easiest change for these viruses to make. This one here seems to be the hardest, and at least in my mind, is probably the most important. And luckily, we haven't seen those changes yet. But sort of keeping in mind that single or double amino acid changes can actually change most of these phenotypes. So, at any given point in time, we're only a handful of mutations away from this virus changing. Of course, what we don't know is how easy it is for these viruses to make those change. We've also seen this figure as well, and that really just to put this in as well to say that even when this virus has infected humans, we haven't seen many of these changes. The sequence in this particular, the first case, of course, did have some of these polymerase mutations, that low-hanging fruit, but nothing else suggestive that there was a whole lot of adaptation occurring in that host. It may have been the fact that it was replicating primarily in the eye, the conjunctiva, which we know in humans is rich in these 2-3 receptors that these avian viruses prefer, and not so much of the 2-6, which this virus has to learn to bind to if it really wants to spread respiratory. And again, looking at the virus loads in this particular individual, most in the eye, not much in the nasal cavity. Also here, just wanted to sort of home on the fact that, these viruses, this is what we call a solid phase binding assay. It's really used to try and figure out if we're seeing receptor changes in these viruses. So here's a range of substrates. We're looking to see both these 2-3 and 2-6 forms. If we look at a human virus here, so this is the pandemic H5N1 of 2009, binds this human form of the receptor, the 2-6, very, very well, and certainly not the 2-3s. If we look at a virus from birds, so 2344B in birds, that at least HA is genetically similar to the bovine. Again, it's the opposite. So it binds these 2-3 receptors really, really well, but not the 2-6. And that's exactly what we're seeing in the bovine isolates as well, where they're recognizing the avian form of the receptor and not the human form. So again, in almost all biological facets, these viruses, the 2344Bs, whether they be from birds, whether they be from foxes, mountain lions, dolphins, or bovines, are still primarily avian viruses in a lot of their genetic features. I'm not going to go too much in that. I'm going to sort of stop here, I think, with this slide. And that's, to me, the take home. This is a virus that is still very much avian-like in its properties. From at least what I'm seeing, there doesn't seem to be a huge amount of pressure in the way this virus is replicating and transmitting in cows for that to change. But I think what we've got to realize is we're a few amino acids away from a virus that could be very, very catastrophic. And we're giving this virus more opportunities to sample new hosts. All of these different hosts provide a different selective advantage or different selective pressure on that virus, driving it to change in ways in which we really don't know. I mean, cows, had you asked me 2 years ago if we would find this virus in cows or in dolphins and porpoises, I would say no, we wouldn't. And of course, 2 years later now, we know it can infect both of these hosts. We have no idea what the pressures are in these hosts to drive change in the virus. But unfortunately, we're finding out, unfortunately, in ways we really don't want to. So I'm going to stop there, hand over to Mike and thank a lot of people involved in these studies. Thank you. And apologies for you having to watch me wildly waffle on with no pictures earlier on. Thank you.
Thanks very much, Dr. Webby. Just before Asante continues, I just wanted the members of the audience to check the chat for a link. That link is to our second poll. And if you could take that poll during the session, we would be very much appreciated. Over to you, Asante.
Thank you, Dr. Douglas. Okay, special thank you to you, Dr. Webby, as well for your presentation. Our next speaker is Professor Michael Ostrom. Professor Ostrom is Regents Professor, Matt Knight Residential Endowed Chair in Public Health, the Director of the Center for Infectious Disease Research and Policy, Distinguished Teaching Professor in the Division of Environmental Health Sciences School of Public Health, and he also wears many professorial hats at the University of Minnesota. Professor Ostrom is a member of the National Academy of Medicine and the Council of Foreign Relations. In June 2005, Professor was appointed by Michael Levitt, Secretary to the Department of Health and Human Services, to the newly established National Science Advisory Board on Biosecurity. From June 2018 through May 2019, he served as a science envoy for health security on behalf of the U.S. Department of State. And in November 2020, Professor Olson was appointed to President-elect Joe Biden's COVID-19 Transition Advisory Board. He is the author of 2 New York Times bestselling books. These are Living Terrors, What America needs to know to survive the coming bioterrorist catastrophe, and his second, The Deadliest Enemy, or War Against Killer Germs. Professor Olson has received numerous honors and has been the recipient of 6 major research awards from the NIH and the CDC. Today, he will speak to us on bird flu, pandemics, and global health security. What are the implications for pandemic preparedness and global health security? Welcome, Professor Olson.
Thank you very much, Dr. Griffin, and to you also, Dr. Douglas. Let me just say it's a real honor to be on the same screen with Dr. Swain, Fry, and Webby. I've learned much from you over the years, and today I'll attempt to reflect on, as an epidemiologist, where do I see us at this point? I don't have slides, as I'm going to try to provide more perspective on what's happening. Let's just back up first of all and remind ourselves what a pandemic is. A pandemic is basically a worldwide outbreak of an infectious agent. And that means by a worldwide outbreak, it doesn't necessarily have to happen overnight, as some would consider HIV a world pandemic when it first emerged back in the 1980s. However, we typically reserve the concept of a pandemic and we think of the devastating impact in a short period of time to one of 2 respiratory viruses. For many decades, we assumed it was largely influenza. And then, of course, with the 2019 emergence of SARS-CoV-2, we now recognize that also can be responsible for a pandemic. But what I'm really focusing on today are, in fact, those agents that I call viruses with wings. And really only influenza and coronaviruses meet that standard today. That could change tomorrow if, in fact, we find changes occurring, for example, to Ebola virus had become a respiratory transmitted agent. There's some hints of that now. Maybe one day that would happen. But I think our focus really is best left for influenza and coronaviruses in general. Now, to remind everyone, when we talk about pandemics, they go back to antiquity. You can go back to early Greek literature and see that Hippocrates talked about the potential for pandemics and surely matching up what likely was influenza at that time in human. But in the modern world of pandemics, really starting with 1918, we recognize that, of course, as the horrible swine flu pandemic, as it was called at the time, with H5N1 recognized in later years in the world of virology that was the cause of that. Then we saw, as has happened with each of these influenza pandemic strains, the virus basically coming in some kind of relationship with humans that was not the devastating pandemic. And so from 1918 on until 1957, H5N1 continued to circulate around the world, all of it having originated in that 1918 spillover. And then it was in 1957 that H2N2 emerged, H5N1 disappeared. That stayed around for almost 11 years when H3N2 appeared, and then H1N1 or H2N2 disappeared. And then, interestingly, in 1977, H5N1 reappeared together now with H3N2, both of them present. And at that time, it was believed that, in fact, the H5N1 appeared from Russia, and the escaped virus from laboratory research that was going on, particularly looking at attenuated vaccine work in Eastern Russia. So we coexisted with H5N1 and H3N2. And then in 2009, we saw another very interesting event where we had, as you know, the new H5N1 pandemic emerged in April of 2009, originating from swine operations in central Mexico and replacing the previous H5N1. And now that is what is with us today, as well as H3N2. And then, of course, as I mentioned, COVID appearing in 2019. Now, I'll come back to this in a moment because there's actually some very important lessons to be learned for our preparedness for influenza from what we just experienced with COVID. But for now, let me just really emphasize the fact that when we sit here today talking about influenza pandemics, there will be additional influenza pandemics. As I often say, the pandemic clock is ticking. We just don't know what time it is. Some would say, oh, well, we just got done with the pandemic. We won't see one happening again soon. Some years will be intervened between. And we can't say that. We don't know. We could be just as ripe for an influenza pandemic in humans today as we've been at any time. What's important, though, to understand is in terms of what could happen in the future, how does that relate to H5N1? And I must say that, first of all, as Dr. Swain and Dr. Webby have pointed out to us here, the H5N1 we're dealing with today, the 2344B, is not the same H5N1 we were dealing with when it first emerged in 1997 in Asia. In fact, while it surely is the progenitor virus, there have been a lot of changes that have occurred since that time. So we have to be careful about trying to extrapolate events that have occurred over these past years since 1997 to now. But I think it is notable, just for everyone to be brought up to speed, what has happened with H5N1 in humans and what that means. And I, along with Dr. Webby, who are part of the NIH efforts in this area, have spent time in Southeast Asia, recognizing that after the 1997 outbreak that occurred in Hong Kong, where there were 18 cases and 6 deaths, and a major effort to eradicate the virus out of the markets of Hong Kong, with large culling operations taking place not just there, but in parts of the Guangdong province. And it was labeled at that time that we had avoided the next pandemic because of that activity. Well, of course, then you know that in late 2002, we saw new cases of H5N1 emerge, again, descendants of what we saw in Hong Kong. And then over the period of roughly 2003-4, right up through 2013, we saw a series of cases that occur each year, largely tied back to the direct contact with infected poultry or aquatic birds. And at that point, we saw anywhere from 100 to 115 cases a year in humans. Occasionally, there appeared to be limited evidence of transmission between people, but very, very limited. And that number from the 100 to 115 cases a year from the 2005-6 time period decreased right up through 2013, when it was as low as 39 cases. Now, I must tell you, and today, listening to our esteemed speakers, Dr. Webby and Dr. Swain, on this issue, I have to tell you, I recall very, very clearly. I can tell you exactly what I had on at the moment. When I sat in a National Science Advisory Board meeting, Biosecurity, in Washington, D.C. As was in my introduction noted, I was a member of that board from its first days in 2005 up through 2014. The NSABB is an organization within the federal government of the United States with a purpose of basically looking at potentially dangerous research that could be result in the creation of more infectious, more lethal viruses or other infectious agents, and more transmissible. And so this was something that we had great concern about, particularly as it related to potential laboratory leaks. In 2012, 2 very prominent influenza experts came to the NSABB and announced that based on their work, they believe that we were literally no more than 1 to 2 mutations away from the then H5N1 causing a global pandemic. And I must tell you, it was a very, very sobering moment. Not a lot changed in terms of how we were better prepared for a pandemic, but at that point, at least, everyone took note. Well, what happened? In 2012 to 2015, the case numbers just continued to decrease in humans. We continue to see bird activity and various levels, but it was really all about decreasing human cases. Then in 2015 and early 2016, we saw a very unique event happen, particularly in the Nile River Valley, for which there were 145 cases, of which over half died, largely associated with duck farming in the Nile River Valley in association with infected ducks. Again, wow, now it's going to happen in 2016. We're just on the cusp of a new major H5N1 pandemic. And then all of a sudden cases disappeared. And literally from 2017 to 2024, 7 years, there have been 33 cases, that's it, as well as 11 deaths in humans. And the lowest activity that we've seen in human-related transmission, bird to human, has occurred in these last 7 years. So from that perspective, as you heard also, the lower level of bird activity before we were talking about the potential for bovine infection made one almost think was H5N1 on its last gasp. Well, of course, we know that's not true in the poultry side of the house. When 2344B emerged, we begin to see the kind of devastation in the poultry industry that we had not seen before, with the exception of limited events in 2015 and 16. So the challenge we have today, does this tell us anything about the future? What does it mean for our preparedness and the likelihood of an event? And I must say at this point, I really want to emphasize something that Richard said. I think it's really important. We may be just several mutations away from a devastating potential H5N1 pandemic. At the same time, we don't know. And I think this has created a lot of confusion, particularly among my colleagues. I'm the one that actually has been publishing on the need to be prepared for an influenza pandemic. In 2005, I had the unique opportunity to author a article that was simultaneously published in the New England Journal of Medicine, Nature, and Foreign Affairs, saying we're unprepared. All three groups came together. And I've not seen us much better prepared for a flu pandemic today than we were basically in 2005. So I want to continue to raise that clarion call, and I'll come back to that in a moment to talk about what I mean. At this point, however, I think it also is fair to say that we don't know that this is going to cause the next pandemic. And I think what we've learned from the public discussion and debate around COVID and what that is that we in public health have been accused often of crying wolf, of saying something's going to happen and it hasn't happened. And I don't know how you could interpret COVID to not be the tragedy that it was, but today there are a number of people who felt that even there, the actions that public health took were not warranted given what happened, despite the fact that over 7 million people worldwide died from COVID. I don't get that. But I think we're in a very difficult position today because there are those that think we are just again crying wolf and that this is not necessarily a major risk. So to me, the important issue that we have to think about today for preparedness for a pandemic is nuancing our message. 1, pandemics will continue to happen. 2, they can be devastating, much more devastating even than what we saw with COVID. 3, we are not prepared for the next influenza or COVID or coronavirus pandemic, and I'll explain that in a moment. And we have to keep emphasizing these messages that should not be confused to a debate. Is H5N1 going to cause the next human pandemic? We don't know. I could share with you, and as Richard just did, some of the data on looking at receptor site issues and so forth. You know, this is another virus that when it's put into swine, we surely can see them getting infected without evidence of them transmitting to each other. There's a number of pieces of data that say we just don't know. So this is a very difficult position to be. If you're trying to convince your government leaders, you're trying to convince the private sector for investment in this area, the challenge is it going to happen or not? Who's going to invest in potentially millions to billions of dollars creating new vaccines that may or may not ever get used? And what does that mean? So I leave you with the fact that accept this uncertainty. And in each case, I think just qualify it by saying this could happen. And should this happen, what does that mean? Well, one of the things that I found myself in a somewhat unusual position, again, I was a bit counter to my colleagues, in February of 2020, when COVID was first emerging as it did, I was very clear in saying we should not be doing lockdowns, this concept of shutting down everything in society. And that is at a time when everyone in public health and many community leaders were saying, oh my, we have to do that. Yes, I was very concerned about what would happen to our healthcare systems as they were being overwhelmed in many parts of the world. and what we could do to reduce that pressure on those. But at the same time, I recognize that a pandemic of influenza could easily last 18 to 36 months, if not more. Are we prepared today to whatever action we're taking to sustain that for 18 to 36 months or more? And with total lockdowns, that was not even possible to do. And so what some of us advise is, for example, create snow days. The idea that if hospitals are becoming overrun, could we have enough data to actually say, at this level of hospitalization or occurrence in the community, we will ask people to refrain from being in public places, reorder work preference, et cetera, at least for several weeks until we see the numbers come back down. Well, I bring that up today because this is a huge challenge we have going forward, is when the next pandemic begins, what in fact are going to be our first marching orders? Will we actually, because now lockdowns have been totally, I think, almost minimized as a potential opportunity, what can we do? Well, we have a number of other what we call non-pharmaceutical interventions, things like masking, particularly effective masking with N95s, et cetera. But again, not going to be the kind of stopgap measure we need. And that brings me to my final point about preparedness. It's all about vaccines, 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 vaccines. Unfortunately, today, we have only made limited advancement in improving on the vaccines of 30 and 40 years ago. When we look today at what our influenza vaccines can do, and particularly taking either seasonal or the experience we had in 2009 with the pandemic, is the fact that most of our influenza vaccine research would tell you that maybe we reduce the issue of illness, serious illness, hospitalizations, and deaths by 20 to 50% in a given year. And that could be expected to be the same with the pandemic. We have no good data supporting the fact that influenza vaccinations, as we currently have them, in fact, significantly reduce transmission or infection itself. And so this reminds me very much of what we had with COVID. COVID vaccines were good vaccines, but not great. What we've learned now is that they provide potentially 4 to 6 months of protection against serious illness, hospitalizations, and deaths, but that even over time, that wanes to the point that 1 year, there may be very limited, if any, protection, even against serious illness and death. And as opposed to being vaccinated, and therefore I can say, well, I had my 2 doses of vaccine 2 years ago, we now know that with COVID, for example, it's all about when did you get your last dose? So that people who have had only 1 dose, but they got it within the last 2 to 3 months, are in much better position than somebody got 3 doses back in 2001, 2002. Well, we have the same sense about influenza vaccines. And the fact of the matter is we need new and better influenza vaccines. Our group at the Center for Infectious Disease Research and Policy, with support from the Wellcome Trust and the NIH, have worked hard to develop what we call influenza vaccine roadmaps that in fact are all about keeping track of the kind of research needs that we need to get new and improved influenza vaccines that are more durable, have a broader level of protection, and a longer endurance of the protection. And so until we have that, we're in trouble. And we're going to constantly be behind the 8 ball. On top of that, will we have vaccines in time? In 2009, when we already had seasonal vaccine capacity around the world, and you may recall, the H5N1 in Mexico basically showed up in late April. By the time, even in our United States, with the access we had to manufacturing capacity, the first vaccines did not roll into our communities until October, and most of it did not arrive, the vaccine, until the following winter. Now, you can say, well, that's still pretty good. The problem was we had 2 waves of the pandemic that occurred in that spring of 2009, and the larger peak that occurred in mid to late September. We missed both of those in terms of protection, just because of the time it takes to make vaccines and to get them out. And even then, again, these are vaccines that did not provide the kind of level of protection that we all would hope for. What did that teach us? Well, it taught us that we still have that problem today, even with mRNA vaccines now available And with the potential to use them for influenza, we're still talking about, months before we're going to see capacity geared up and get those vaccines out. Now, one of the challenges with that, of course, is the fact that we talk often about how long does it take to get a vaccine from original concept and research and development to manufacturing and distribution. So how long? Well, I always say it's not from the time from the decision to make the vaccine to the first dose. It's about the time to the second dose, the last dose. Who is it that gets the last dose? And we saw globally with COVID, for example, that many of the low and middle income countries didn't have really ready access to vaccines until well into the second, almost going into the third year of the pandemic. And so we have a lot of work yet to do here. So from a government standpoint right now, we need the governments of the world to come together to recognize why we need really a crash effort to improve on the influenza vaccines we have today. And while they're doing that, bring along the coronavirus vaccines with them. Until we see that, we're going to continue to be highly vulnerable to the next pandemic. And I see a number of my colleagues today talking about the importance of surveillance. If we don't have surveillance, we're going to have a pandemic. Well, that's just simply not true. I am an epidemiologist. The two four-letter words in my life are love and data. I want data. I find that to be very important. So I want surveillance data. But if we tested every cow every day in this country, that wouldn't guarantee that one night there wouldn't be a reassortment or mutation changes that resulted in the new pandemic. So we have to understand, if it's going to happen, it's going to happen. We can't stop it. What we have to do is be prepared to respond to it. And that's where I think we're sorely missing. Finally, let me just say a few words about antivirals. In the absence of vaccines that are effective, I think that this is really an important issue of what can we do in the interim before vaccines might be available, and even after they become available, how can we work on that issue? Well, we've already realized as a global population, with the amount of antivirals that we have for influenza, as well as for COVID, they're extremely limited, very limited. And even today, when we look at oseltamivir as a neuraminidase inhibitor, we're seeing rapidly increasing levels of antiviral resistance to oseltamivir, with challenges as to what that might be. We have no major new efforts globally right now to bring us new improved antivirals, or even so, if we could bring them forward, who would manufacture them in the quantities at the time of the pandemic? So I leave you with the fact that, number one, I don't know what's going to happen. You got people on this webinar a lot smarter than me that can tell us that. I don't know. I do know that H5N1 is surely a challenge that we have to look at. It's been a challenge that has surely taken me to a place in my career over the last 20 plus years that have left me really concerned, if not outright scared sometimes. And I don't know what to do today, what it means to be today, but it's there. The next thing is, but even if H5N1 doesn't do it, we're going to have another influenza pandemic, and it could be just as bad, if not worse, than 1918. And so I think this is a challenge we have. So I want to thank you for the opportunity to be with you here today. And I hope that these words of perspective are helpful in trying to interpret the current public health priorities we have for pandemics. Thank you very much, Dr. Douglas.
Thank you very much, Professor Osterholm, and thanks to all of the previous presenters who have given us a really sharp perspective of what's ongoing on the H5N1 panzootic as we see it evolving across the globe, and particularly in the USA. Now we turn ourselves to the Q&A section. We had one question which was answered by Matthias, so we won't go through that one. There is a question, however, from Lisa Musai. You want to say something, Dr. Swain, before?
Yeah, that's I was technologically challenged and I couldn't answer that question, and great that Dr. Webby put in some information. But just to add one little piece to that, and if you haven't read the answer, Dr. Webby said there's really no genetic changes that would explain the dairy cattle infection. And he thought it was mostly a chance occurrence. And I'll say there's a little bit of circumstantial evidence or some early experimental evidence that would additional support that statement by Dr. Webby. One is just last week, the German research group FLI reported on their initial experiments where intermammary exposure via injecting the virus up the teat canal with the U.S. bovine isolate produced infection in those mammary glands of the cows, but also they used a second virus in other cows, which was a wild bird virus from Germany, and they could reproduce the same infection. And these viruses, although they have 4 genes in common, gene segments in common, there are 4 that are different, which would suggest that at least that 4 genes that are from Eurasia are by themselves sufficient to produce a dairy cattle infection. The other piece of circumstantial evidence, and this is some work that Dr. Webby's group, Dr. Webby specifically working with Lars Larsen in Denmark, looking at receptor binding, found that in the mammary gland there are receptors for both alpha-2/3 and alpha-2/6 influenza viruses, and then also there are receptors deep in the lung, which would suggest that initial case had to probably be somehow an exposure deep in the lung of a cow, and how that happened no one knows, or somehow exposure to the mammary gland, which becomes even more difficult to kind of visualize a wild bird exposing a mammary gland. And also there was a recent paper published in Emerging Infectious Diseases that showed a very similar receptor binding. So those 3 things, what Dr. Webby said about there's no significant genetic change that could explain it, and these other 2 pieces of circumstantial evidence, the receptor binding, as well as the 2 different virus strains that produce infection experimentally, would suggest it was sort of a chance occurrence.
Yeah, all very intriguing, and as I think.
Could I also add to that? I think, David, supporting that very point is also our colleagues in Canada and Europe have not yet reported any positive milk samples, suggesting that we're not missing it either in these cattle. And the fact that there was very limited movement of the cattle from the panhandle of Texas to Canada, which of course you would think that in fact, if it could infect us here in the United States, it surely should infect cattle in Canada. And if it was a cattle movement, that was really important, in that would also explain this as a kind of a one-off kind of thing, that it is not happening widespread in Canada or Europe.
Those are all very interesting, and I looked with quite a bit of intrigue with the map that Dr. Frey had, and seeing the pathway in terms of the spread throughout the different states. And I'll reserve that question because I want to skip over to Dr. Musai's question that she's put into the Q&A session. Then I'll follow up. And Dr. Musai asks, what is the proposed effect of climate or environment of the Caribbean to the survivability and pathogenicity of the HPAI virus? So I guess that's a question of, will this H5N1, if it unfortunately makes its way to the region, be able to survive our climate and our environment? Any thoughts?
I'll have a crack at that first, and then I don't know if David, you've got any perspective from the sort of the more poultry side. So what we do know about flu is that humidity and temperature are, of course, big drivers of activity. I think there's probably some details that we don't know about that. So high, at least in the context of sort of respiratory transmission, high humidity is not ideal. And flu is an envelope virus, so it is susceptible to increased temperatures, desiccation, etc. So at least in some perspectives, you could make an argument that perhaps in the sort of the Caribbean region, there's it's not ideal conditions for a virus to spread, but of course we know that seasonal flu comes through every part of the world. The timing is just a little bit different. So I certainly wouldn't hang my hat on the fact that this is the conditions are not going to be conducive to the virus spread. I mean, if we're talking specifically about cows as well, again, I think all bets are off the table for that one. This virus is probably transmitting by some sort of mechanical spread, where I don't think humidity and temperature have much role to play at all. I don't know, David, if you got much to add from sort of a poultry perspective or not.
Yeah, I'd have to say not necessarily about the Caribbean area, but globally, I would say, climate change has had an impact on avian influenza, mainly by changes in migratory flyways and the length of time birds are for example, in the Northern Hemisphere in the summer breeding grounds, and because of the warming climate, they can go further northward and they can distribute wider in those grounds. So there's seems to be more lateral transmission or peripolar transmission going on among the different wild bird species. So that could impact the flyways, the way the viruses move within those flyways, and it could change the prevalence of the virus in those flyways, which all could have an impact in the Caribbean region, but it's more at a global level.
Yeah, and I would add to, in light of that, with the, well, El Nino and La Nina, the switching of the two cycles, we will get more intense storms. And that is a factor within the migratory bird flying down south to South America. And usually what we see anecdotally in Barbados is that whenever we have stormy weather, the birds actually, what they say in colloquial language, they come down. They actually rest in the islands before making the journey down. So the more storm activity you have, then the higher the risk you're going to have these birds coming down into the island. So that is something that we should be cognizant of and aware of as well. And hopefully we do have some persons from Latin America as well on this webinar, or who will see this in the future as well, because we're recording it. I wanted to pose a question to Dr. Fry, and this is in relation to the bovine influenza within the outbreak that we have. This is something I'm not sure we do have an answer at the moment, but maybe to get her train of thought in terms of the movement of the virus through the USA and what could be the mechanism or mechanisms. I would assume it's something mechanical, whether, you know, the others are being abraded whilst the machinery or the hands of the persons who are doing the milking are contaminated. But what are your thoughts on that?
Yeah, actually, there was an interesting publication from the USDA. I'll put the link in the chat after I answer this question, where they talk about the transmission in Michigan from dairy cattle to poultry. They looked at, there were some employees that worked either at a dairy farm and a poultry facility, or employees that lived together, where some employees worked at a dairy farm and some worked at a poultry facility, which I thought was very interesting. Also, vehicles being driven between facilities. So that seemed to point to fomites. I kind of put that picture of the traditional US dairy parlor in because knowing how much virus is in milk, I just would think that the parlor would have to be a place for transmission. I think, like you said, I don't think people know exactly if it's aerosolization of milk in the parlor going cow to cow, if it's aerosolization of nasal secretions going cow to cow, or if it's direct contact with the milk, because yes, those claws are attached to the cow, they come off, the next cow goes in and they're directly attached. There's not a cleaning process happening in between each cow. If you can imagine milking, 10,000 cows, you can't clean a unit between each cow. So I don't know which one it is, but I would suspect because there's so much virus in the milk has got to be playing a factor. And then the cats drinking raw milk and then getting it and dying, also very interesting. And I've heard people talk about, maybe the colonization in the lymph nodes in the pharynx as a route of entry, not the GI tract, but just that exposure orally to the respiratory tract and things. So I think there's a lot of potential pathways, unfortunately. I don't know if anybody else wants to chime in and answer that as well.
Can I just add, this is Mike again, I'm somewhat surprised by the number of people who are surprised that cats are involved right now. I think any of us that recall the 2003-2004 activity in Southeast Asia recall that, in fact, when they culled the chickens and then fed them to the tigers at different zoos in Thailand, all of them got infected and died. And, that surely was an early evidence of consumption. And I think, David, as you talked about, the kind of mammals we're seeing today that predate on potential dead birds, et cetera. I think that this is not surprising that we're seeing it not just in cats, but also other animal species. I think the mechanism by which it actually happens isn't clear, but surely ingestion of these dead animals is still capable of causing transmission to the consumer of those dead animals.
Yeah, I was also just listening to an older webinar on the CDC about early seal outbreaks, and that seals don't typically actually eat birds. So they were talking about some of their behaviors to like roll in dead bird carcasses, or maybe there was feces from the birds in the water. So I think I thought naively that it was just carnivores consuming birds, but there probably are multiple routes.
Yeah.
Yeah, and with the recent elephant seal deaths off the coast of Argentina, it would definitely support other than consumption of dead birds. The number of die-offs in those colonies, especially among the very young pups, which is before they would actually do any consumption outside of mother's milk.
Oh, very intriguing, to be quite honest. This is a question for you, Richard. What animal hosts would you say poses the highest risk for pandemic potential? Would this be pigs because of the dual receptors that they have, 2.3 and 2.6? Or at this point in time, there's no higher risk for one specific animal host?
Yeah, so I think it's a great question. I think the answer is probably it is pigs. But that's because we know pigs can do that. I think as I tried to get across, there are many hosts that are getting infected by this virus that we have no idea. Are they more pig-like in terms of potentially driving this virus to be more able to infect humans, or are they sort of just like another bird where there's really no pressure on that virus to change, replicating within it? So yeah, we can say pigs are that's a host we really don't want this virus in. Because we do know it has both receptors. We do know avian viruses can get into that host, and over the period, over the course of time, can pick up sort of more human virus-like traits. Unfortunately, for most of these other species that are getting infected now, we just don't know.
So has there been any studies looking at H5N1, particularly in the pig hosts, to see if there is, the potential for reassortment, or, how the virus would actually evolve within the pig?
Yeah, so and I think Mike alluded to some of these as well. There have been a number of sort of experimental challenges of pigs over the years with these H5 viruses. Early on, they really didn't do much. So there was a little trace amounts of replication, if any at all, and certainly no transmission. I think more recently, Amy Vincent and her team at NADC, part of the USDA here in the US, Jürgen Reed, Kansas State, have put some of these more recent viruses into swine, and there has been a bit of replication, and a little bit of transmission as well. It's far from robust, so this is not a virus that is going to, if it gets the opportunity to go in pigs, is going to immediately take off and be like a normal pig influenza virus. But it can get in, it can replicate, and of course, that provides an avenue for further adaptations to occur.
Okay, I see Dr. Trotman has his hand up. Come in.
Yeah, thanks, Kirk. I just want a quick question for Dr. Fry. In your work, have you identified or isolated the virus in the cattle tissue? Because one of the big questions that most of us in the regulatory field are looking at is the implications for international trade and the movement through tissues, cattle products from country to country, and whether that can play a role in the introduction and spread of the virus through that route. I don't know if you can answer that.
Yeah, there was. I hope I'm up to date on this. There was one. The FDA looked at. Sorry, maybe it was the USDA looked at 109 carcasses that were going for slaughter. One carcass did have H5N1 detected in the diaphragm muscle, but it was, I think it was just viral particles. I don't really have the details on that. And then there has been one cow, when animals go to slaughterhouses and they're USDA regulated, there are FSIS employees, there are like food inspectors there, veterinarians who are trained to detect lesions in the carcasses and then pull them out of the line. And so there was one abnormal lung tissue that was H5N1. So those are the only two instances I know of H5N1 in carcasses. And then, there has been a lot of work to prove that pasteurization would kill the virus in milk, and then cooking would also kill the virus in beef. Does anyone on the panel know of any other more up-to-date information on tissues?
The only thing I remember was the paper by Burrow et al. At EID had positivity in kidney. Infrequently, but positivity in kidney. And I think rumen contents also had some positivity, but very high CT values, low virus content.
Mm-hmm. I'll put the link for the diaphragm muscle and the lung tissue.
And those make sense from what Dr. Fry said earlier, looking at the samples from that multiple diagnostic lab case where, you know, the most positive samples, both titer and number, were in milk samples. And then on down the line was respiratory samples, and then also urine, in which those were nasal swabs, I think you had in urine. So, you know, possibility of kidney and also lung would make sense that you would have some positivity. Although the CT of the amount of virus is much, much lower than milk.
So did that answer your question, Dr. Trotman, or did you want to? Dr. Trotman? Oh, you're still muted, sorry.
Yes, thanks. It does ask my question. I'm not getting the impression that we found any virus in muscle tissue, which is the main concern that we would have with respect to international trade. But I take the point that there's obviously still a lot of work that needs to be done to see exactly what's going on with the virus. But thanks a lot.
Okay, sure, no problem. Question for you, Dr. Swain. Given the recent developments in Finland, where they're vaccinating humans at high risk on mink farms within Finland, what is the scope for bovine vaccination and even farm worker vaccinations, you think, within the US at present? And then if this unfortunately escapes and goes somewhere else, that to occur as well?
Yeah, I mean, that's really a question on the public health side. And, you know, there are discussions in the US about dairy farm workers, do they need to have vaccination, or other high risk people who may be in high risk occupational series, which could potentially even be people who do depopulation of poultry flocks that are infected. So those are all, I guess, critical issues for discussion within that public health arena. Is, you know, how do you protect the dairy workers? Is the personal protective equipment sufficient to protection? You know, the 3 cases that are positive were before there was widespread knowledge about the dairy infection. So were there any PPE used by them? The answer may be no, or it may be yes. So those are all public health questions. I think that's all I could say.
Okay. But in regards to vaccination of herds or anything, you don't think that's practical, or?
So of course, it depends on the country in making a decision on, you know, what types of birds and where, et cetera, should be done. You know, in most of the high income countries right now, vaccination is not being pursued in poultry. And of course, in most of the high income countries, whether it's the United States or the European Union, Canada, or even Australia, for the most part, with the 2344B, most of those cases are coming from environmental exposure from outside the barns into the barns by human activity. And it's not very many of those cases are lateral spread. So if you could go back and look at the paper in, I think it was late 2002, which compiled all the first cases in poultry, 87% of those cases, based on the molecular sequence and analysis, suggested primary introductions from the environment of the wild birds. And only 13% could have been lateral spread. And that's pretty well been identified in the European Union also in their case studies looking at that. But that's not necessarily true when you look at medium and low income countries where there is more live markets and value chain movement of birds and fomites. So there is actually more lateral transmission from farm to farm than there is probably wild bird introduction. But vaccination is very effective if done correctly. I think Mike would really understand, and tout this, but back in the early 2000s, we had a major number of cases in Vietnam of human cases, and they implemented a vaccination program. And the next year, the number of cases declined from around 50 to about 8, and those had epidemiologic exposure to non-vaccinated poultry. So if you can reduce the infection in poultry, you reduce the human exposure in those live market systems, and that has typically shown reduction in human exposure and human infections.
We know that there are also trade implications for that, where export of poultry, etc., occurs. So it's definitely a decision that weighs heavy on those who have the remit and responsibility for the animal health in those areas. And given that you touched on the situation in Vietnam at that point in time, and also lower to middle income countries, I wanted to steer a question over to Professor Osterhall. And this is in regards to the ongoing outbreak that we have. What is the role of migrant worker health in the ongoing influenza outbreaks on farms? And what can you speak to in terms of the vaccination and the global health security measures that can be taken?
Well, let me, if I correctly understand the question, let me just address the issue of migrant workers in the United States. This has posed a real challenge for us because, frankly, many of the operations that we've been involved with, undocumented workers have played a key role in keeping those operations up and alive, and they're invaluable employees. The problem is once the U.S. government, whether it be public health or other, come into those farms, we've had real challenges, and I think Dr. Fry and others can talk to that, of trying to enroll those employees in any kind of follow-up. They're fearful for their status, and this has created a major challenge. I've had contact with a number of state health departments where they've tried to do outreach, and the suspicion and fears have been a big barrier. And so I think this is a huge issue. I think the vaccination issue is another one that's a challenge in that you make it voluntary, and how well would you have compliance? And you make it mandatory, which has all the challenges of what we saw with COVID. And then in addition, people will say, well, you've had 3 minor illnesses so far. Would you vaccinate large populations just because of that kind of human experience? And, I'm not suggesting you don't, but I think that's another issue that come up. So I don't think it's clear cut. I will say that from a standpoint of, and this goes back to my own days as a young boy milking cows in Iowa, I can tell you that, the kind of PPE we're talking about, the idea of an N95 respirator, goggles that fog up, et cetera, has made the use of PPE in these dairy areas very difficult at best. And we've never really designed equipment that is user-friendly and that is effective in those environments. So, the feedback we've gotten so far is that the PPE is often considered a greater hazard in terms of fogged up glasses, et cetera, than not using it. And so that I think we have a lot of work to do in this area to try to find the most effective ways. No one yet has really figured out, I think since the advent of electricity, how to milk cows better. I mean, we've surely improved on it, but I think, there's not a way yet that we can minimize, I think, the kind of aerosolization that occurs with that type of operation.
Thanks very much. And I know, and there is that point too, because I know even within the Caribbean context, we do have some emerging and ongoing issues of migration. In particular, I could think of Trinidad and Tobago and Venezuela, where we have migrants moving from Venezuela over to Trinidad. Some of them are transitory. They also move livestock, and they also move wildlife from Venezuela over to Trinidad and Tobago. And I know I saw Dr. Muse on the call before. She's from Trinidad and Tobago. So I am thinking that this might be something of concern for them in particular, but also for the rest of the region as well. And before I go, I know that you had asked a question, Dr. Frey. I wasn't able to give you an answer, but perhaps Dr. Trotman might be able to give you an answer. I think Dr. Frey was asking about the cattle population in Barbados and what that makeup was in terms of beef and in terms of the milk producers. She wanted to get an idea of that.
Yeah, the majority of the cattle that we have in Barbados are dairy cattle. Our primary cattle industry is dairy. Very small beef industry. We don't have the land mass for a beef industry. So almost exclusively cattle or dual purpose animals. Not a lot of animals, but relatively speaking, for the size of the population in Barbados, we are relatively self-sufficient in fresh milk. So it's a significant, although numerically compared to large countries, it may not sound like much. Relatively speaking, it is a significant contributor to food security. So it's mainly dairy and fresh milk. That's what we have here. So obviously there are concerns about importation of live animals, first of all, to replenish our dairy herds. Milk is secondarily. Milk obviously will be pasteurized, so I don't think it's a major risk in the importation of milk. But live animals, especially, we are very concerned about the importation of live animals that may be carrying the virus within them, and then being spread to the local cattle population. So that's where we are.
Yeah, I just saw one question someone sent into the chat. And we do have, probably this is the last one we will take. How practical is it to vaccinate poultry flocks in the region if the disease threatens? And I guess this is probably a question for Dr. Swain, since this is right in his wheelhouse.
Yeah, and the FAO Food and Agriculture Organization has just launched a new virtual learning program, which is about vaccination stewardship. So yes, vaccination can be done, but it really should be risk-based. So determining what is the risk for poultry within the country or a region in the country. And based on that, whether it's surveillance of wild birds or in poultry in adjacent countries, then you need to make a determination if vaccination is appropriate. And vaccination can be effective. It's been shown to be effective in multiple countries, including the 6 that have just started vaccinating in 2023. But they didn't vaccinate all the poultry. They vaccinated the ones that were high risk, and that were long-lived poultry. Mainly it was layers and breeders, heavy and light breeders. So it's always risk-based, and it fits the situation of the country based on that analysis.
Thanks very much. Oh, that's something that also I forgot to mention. In terms of not only bovines being infected with H5N1, but we also saw on a few farms goats being infected as well. And I'm not sure how that fits into the grand scheme of the risk spectrum for H5N1, and should we be concerned that goats are being infected as well? I think this is probably where the goats were housed with poultry, or on the same farm where you had infected poultry, or maybe even infected cows as well. Would you be concerned that we're seeing infections also in other livestock animals, such as goats? Because we know in the Caribbean, we do like to have a herd or two of these goats as well.
That's a very good question. It's funny. I was just writing in the chat. I didn't add it in my slides, but so a week before the dairy cattle announcement, there was a goat. I'm not going to say a herd. A few goats living with backyard poultry. The goat kids developed. I think they were neurologic, and they got H5N1 from the poultry. That strain of the virus was not linked to the dairy cattle. I'm not sure. I don't remember I don't know if it was a clade difference or if anyone else can comment, but I don't remember. Just know it wasn't linked to the dairy cattle. And then more recently, there were some alpacas that were living with backyard poultry in Idaho that also had H5N1. They were asymptomatic, and the strain that the poultry had was from the dairy cattle, but it was not. There were no dairy cattle on the premises where the alpacas and the poultry were. I think that we have not looked at small ruminants very much, so that is definitely an area of interest for me and some others. There has been discussions about testing more cats, not just cats that are on dairy farms, but just cats in general that die of a neurologic disease and are rabies negative. So some of that I think is starting to happen. But does anyone else on the panel remember if it was a different clade with the goats?
Yeah, it was a different genotype. It was B3.6, which differs from the B3.13 by nuclear protein gene reassortment. But the alpacas is a B3.13. Is that right, Richard, from what I remember anyway?
Yeah, yes, I think so. And that B36 genotype was probably, it's a little like reading tea leaves, was probably the most dominant one in wild birds at that time. So, probably fairly widespread in birds and spilling over into that farm, which I think Mike can comment on, was a pretty unique environment as well.
Mhm. Yep.
Well, I see that we are approaching the time to wrap up. So let me begin first by thanking all of you, Dr. Swain, Dr. Webby, Dr. Frey, Professor Osterholm, Dr. Trotman, also to you, Asante, here at CBS. And we are also very, very happy for all of the persons who joined this webinar live and those who will also take in this webinar in the recorded version of it. We, on behalf of the Center for Biosecurity Studies at the University of the West Indies, we want to encourage you to stay tuned to our webinar series. We will have a very interesting lineup for you as we go forward with the rest of the semester and the rest of the academic year. And we also want to indicate that we have successfully gotten our postgraduate programs approved, and we will be shortly issuing our marketing strategy and getting out there in terms of the biosecurity and biorisk management program that we are launching. We want it to be multidisciplinary. We want it to be engaging and definitely addressing the biothreats that are posed not only to the region, but also globally as well. So on behalf of the entire team here at the Center for Biosecurity Studies, thank you all very, very much. Take care and goodbye.